Welcome back to Flagship Studios. We're here with Steve Harr from Sana Biotechnology, presented yesterday on the main stage at JPM. What did you talk about in your presentation yesterday, Steve?
Well, first off, thank you, Christine, for having me. And we focused on a couple of elements. One, when we set out the company, our major goal was to overcome a couple of the challenges that really are holding back the promise of engineered cells. And we've invested and made progress in two.
Okay.
And one of the most exciting things for us is after years of investment, in 2023, we're now at the place where we're treating patients, and we will understand, does the exciting biology that we've seen in preclinical models really translate into humans? And so we outlined that we recently filed an IND for the first program, which is a hypoimmune or cloaked CAR T cell,
okay,
targeting patients with certain blood cancers. And I think it's really easy to tell, or would be really transparent, does what we do work as we expect it will? Are we able to hide these from being killed by the patient's own immune system? The second, and we hadn't discussed this before, is one of our goals is to be able to cure type 1 diabetes. And we're developing a scalable hypoimmune stem cell based program. We wanted to learn faster, and so we're going to go forward very shortly with gene edited primary islet cells that we will allogeneically, that we will transplant into type 1 diabetics. And you'll know, without any immunosuppression, whether or not what we really have shown in non-human primate and other models translates into humans. And if we have, with the blood cancers, we think we have the possibility of being able to cure, not just treat, tens of thousands of patients per year. With type 1 diabetes, which affects millions of people, millions of people, we'll know at the end of this year. Right now, we believe that a cure for type 1 diabetes is possible. If this works, it becomes inevitable. And that is transformative, because it's going to be hard. It's going to be way harder than we think. And so having that inevitability gives us the resilience to really make it happen.
And that's a huge leap. We're talking about bigger leaps this week. That's a huge leap, the inevitability of curing diabetes. But there is so much always going on at Sana, it's hard even for us to keep up. Beyond what you presented yesterday, give us a sense of some other things cooking at Sana right now.
Beyond what you present, the thing that I'm really excited about is we've been working, that we haven't talked a lot about, at least yesterday, is the ability to deliver gene modification material specifically to certain cells in the body. What that allows us to do is gene-specific control of a cell in a cell-specific way. And today, you can do almost anything you want to a cell in a petri dish. Right? The hard part is delivery. Right? And if we can actually deliver all of that material to the cells we want to get it to, that's transformative. We can't do it to every cell right now, but there are cells where we're getting this really to work, and we're showing that we can knock out genes, we can edit genes in a specific way just in the cells that we want to target. And that, we think, could be really transformative. It's not going to be in humans this year, but it's not that far away.
And what are some of the things you think Sana's working on that the company's least associated with?
That we're what?
Least associated with. What do people not know about Sana in terms of what you're working on?
Well, it's always hard to know what people know.
What do you get asked about last maybe?
Yeah, I think the program that maybe we never talk about that could be really transformative is, what's become clearer and clearer is that, first of all, neurologic diseases are complex.
Yeah.
Right? Second, while most of the symptoms that we think about come from not having properly functioned neurons, most of the diseases are actually diseases of the white matter, right? And the white matter is what really is the soil, which allows the seed and the flower really to flourish, and that fixing that white matter can be transformative. And so we've been working really hard on these glial progenitor cells, which can go into the brain.
What kind of cells?
Glial progenitor cells.
Progenitor cells.
So they're bipotent cells, and they can either become astrocytes, which really are a big part of the support mechanism, or they can become oligodendrocytes, which are kind of the sheath that allows the electronic signals to work in the brain. And just it's context dependent. And if we're able to really transplant these cells, and they migrate throughout the brain and really fix broken cells and make them healthy cells, the number of neurologic conditions that are that we can go after and hopefully really transform it's hard to count. It really is. Now, these are really complex cells, and our scientists have been working on them for a long, long time. We've made a lot of progress. We are at the point where we're really, we're looking forward to an IND next year. And so again, that's one of these things where we never talk about. That's kind of on the pipeline chart, but they could really go after a host of diseases and with really grievous outcomes, and hopefully have a really big impact for these people.
Let me ask you about culture. How many employees now at Sana?
About 425.
You talked about learning faster a few moments ago as part of how you work. How do you inculcate that in a culture? How do you build a culture that learns faster?
Yeah, I would say, first off, well, I always say I have 5 jobs, and one of them is eye and culture. And if culture is not any good, it's my responsibility, it's my fault. If it's to be really great, though, everybody has to buy in, right? That's part one. The second is we spend a lot of time thinking about how do you operationalize a great culture, and what are the attributes of how you actually work together and work that makes it such that you have a great culture. Because to me, a great culture is this undefinable thing that is really actually, do you unleash human potential or do you hold it back? Right? And that great culture to me is one that unleashes human potential. And the next part is who you hire, and that's where you really begin to get into how do you create a culture of learning. I think there are two.
Yeah, what do you hire for?
Yeah, the 2 attributes you cannot compromise on are you have to have people who are intellectually curious, right? If they're not intellectually curious, because what we're doing is we're on the sphere of the tip of innovation, right? And we're learning all the time. We're failing all the time, right? And so it's really will be measured, not often, our success is not often going to be held back by success or failure of a single experiment. It's how fast do we learn. That will define the company that really transforms the field, right? So you have to be intellectually curious, and then we really test for resiliency and grit, because
resiliency and grit.
So those attributes, intellectual curiosity and resiliency/grit, are the uncompromisable attributes of an employee. We actually have specific people who are set aside as cultural bar raisers who interview just for those characteristics for every employee before they're hired at the company.
That's amazing. Yeah. My last question for you has to do with our theme here at JPM, bigger leaps. When you think about, you've talked about so much that's incredibly promising on the horizon at Sana. When we're in this chair in 2028, talking about your JPM presentation, what will Sana be talking about? What bigger leaps will you have been taken between now and 2028, say 5 years out?
Yeah, I will talk about the big leap that I hope will be taken first. If we've truly figured out how to hide allogeneic cells from rejection, you've completely changed the way that we approach so much of medicine, transplant medicine, cellular medicine, and we're going to be spending a decade plus figuring out where are all of the best places for us to apply it ourselves, and where might we be better off partnering with others. And so my goal is that the first thing is in 2028, we're talking about, my goodness, you've solved that. Because if we've solved it, then I believe that first off, hematologic malignancies will be like hepatitis C. The vast majority of patients will be cured with a single treatment. Second, we'll be saying that you guys, like you're launching now, this cure for type 1 diabetes, right? And the third thing you'll be saying is, okay, what's next?
Yeah.
What other cells are you going to be in? Like, that's the bigger leap, is we'll have decided what else is coming. But right now, that's as far as we'll go, but we'll have decided what's next.
From hiding allogeneic cells to cancer to taking diabetes from impossible to inevitable to cure. Big leaps with Steve Harr from Sana Biotech. Thanks so much for being here, Steve.
Thank you. Appreciate it.